I'll check on the dispatches also.
Suman, Kinjal, and Avinash. Let's see.
Angel is there.
There. Okay. I don't see Suman yet.
Mm.
Can we let people in? So, Arushi, everybody in?
Yeah,
Everybody?
I'll let.
Hello, everyone. Good morning. Hi. Hello, Ashutosh.
Yeah, hi Krishna.
Hi. Hi, Parveen. Hi, Salim. How are you?
Hi
Good to
Krishna.
see you all. Good to see you
Good
all.
morning. Good morning.
Good morning. Good morning. So nice to see you all up and early. Yeah. And welcome to PhD Cafe. For our speakers and audience are slowly joining in. And I'll introduce you to Team India Bioscience. That's my colleague Arushi, Ankita, and Rutuja at India Bioscience. Yeah, welcome.
Thank you. Thank you.
So a brief introduction and then get started with the speakers. First speaker is Suman. I don't see her yet. But yeah. The next speaker is Kinjal. Kinjal is there. Great. Great. So as this is day 4, um, Salim, Ashutosh, and Parveen. So basically, as expert mentors, you have to listen to the 20-minute talk, and then you have 10 minutes to give suggestions, feedback, question, answer, just to better their work. It's like a pre-defense experience is what we are trying to give them. And then if we have additional time, we can move into audience questions. But typically, we are not having additional time, and the mentors have been really giving wonderful feedback, which I'm sure even you all will. How can they better this work for their degree completion? Yeah. Any questions, let us know. And my colleague Manjula from India Bioscience has also joined. We are also on YouTube. We're also on YouTube, yeah. Excellent. So, Suman, are you here? Rutuja is the first speaker here. I don't see them. No, I don't see Suman. So, okay, not yet. Kinjal, you are here, right? Kinjal, I see you in the meeting room.
Yes, correct. I'm here.
Well, we will slowly begin, but I'll just give you a heads up. You may need to go first. Would that be okay?
Uh, yeah, I should be okay.
Okay. I understand it's a bit unexpected for you. We'll maybe give it a couple of minutes and then I'll start. And so if by then Suman doesn't show up, then yeah. All right, I think with respect for everyone's time, we'll slowly get going. We will just keep an eye on the chat window for Suman and the meeting room.
Sure.
Welcome everybody. Hope you can see and hear us and see some slides. We are on day 4, session 1, 2, 3, 4, 5, 6, 7, session 7 of PhD Cafe, and our session is membrane biology. So great to have you all here. And we'll have people filtering into the meeting room through the session. So today's session has 3 speakers, Suman, Kinjal, and Avinash. And welcome and all the very best to you. General housekeeping, see us, hear us, use the chat window to add in your questions or suggestions or comments. After every PhD speaker talk, which is 20 minutes, we'll have 10 minutes of Q&A and feedback from the expert mentors who are here. And then if we have time, we'll take audience Q&A. But the audience can keep typing questions in the chat window. And of course, feedback should be constructive and better the work. Thank you to our mentors, Parveen, Salim, and Ashutosh. It's great to have you. Thanks for making the time for this. I know you all have very busy schedules. Thank you so much. We look forward to this session. And the audience has incentive to keep the chat window active, because what we've been doing through all our 3 days of sessions is the most engaging and interactive audience member gets a physical copy of our latest e-compendium. It's getting past the finish line. And you see, we've done a play on the word finish. And challenges and interventions to better support PhD experiences. And this e-compendium has been put together by my colleague Ankita, who manages science communication at India Bioscience. So hot off the press, it's just recently been published and printed. And our most engaging audience member gets to win a free physical copy. So keep the chat window active. We've already given 6 e-compendia so far. Yeah, great. Okay. Then after the session today, which will probably be ending early, we will all do other things that we need to take care of in life. But for the PhD researchers here, we have an open networking from 1 to 2 with Shravanti, and she will be fielding open questions on what's it like to get a postdoc position out of India. How do you start? How do you send emails? Whatever it takes. And this is a different meeting link, and our PhD researchers have been coming there every afternoon from 1 to 2 p.m. So just follow that meeting link and see you there with Shravanti and your lunch. Okay, let's do a desk lunch if possible. All right. So let's get beginning. Let's get started, and no better time to get started because I see Suman in the meeting room. Suman, that's great.
Yes, ma'am.
That's perfect timing, Suman. Great. So welcome to PhD Cafe. We can see you. We can hear you also. Can you start sharing your slides, Suman?
Yes, ma'am. Yes, ma'am. I'm sharing.
And Suman, just to get you up to speed, our expert mentors are here. Ashutosh, Parveen, and Salim. And then all of us are the audience. You have 20 minutes. So my colleague Manjula will keep time if that's possible. So Manjula, 20 minutes starts now. And Suman, after that, there'll be 10 minutes of Q&A.
Okay, ma'am. Thank you, ma'am.
Welcome. All the very best.
Ma'am, I'm audible?
Yes, you are.
Thank you, ma'am. Thank you.
Suman, you might want to go into slideshow. Yeah.
Yeah. Okay, sorry for the delay. So I sponsor integrated PhD scholar working under the guidance of Dr. Rajendra K. Mutiani. So my title of this project is ability to mediate intraocular calcium dynamics triggers nanophagy to moderate pigmentation. So first started with the pathway of skin pigmentation, in which when sunlight falls on our skin, that is keratinocytes, it causes DNA damage and it causes the release of propionanocotin. This propionanocotin hormone cleaves and forms alpha melanocyte stimulating hormone. These alpha message then forms AP3 signals. These AP3 signals then bind with the AP3 receptors. This causes the release of calcium from the endoplasmic reticulum. This release of calcium is then sensed by the stimulant print on the ear, is then oligomerized and then it interacts with the adenyl cyclase 6. This causes the release of cyclic AMP. This cyclic AMP then then stimulates protein kinase A. Then protein kinase A phosphorylates CRAB and CRAB is a transcription factor. It transcribes MITF. MITF is the master transcription regulator, and it transcribes key melanin genes, that is district as NGP100, and this causes a function of pigmentation, and this pigmentation then goes into the keratinocytes, and we found the pigmentation on our skin. So in all this process, the role of IP3R in pigmentation is still unknown. So to discover this, we started with our own lab microanalysis, in which the lightly pigmented primary melanocytes, when stimulated with the tyrosine, which is the pigmentary agent, we saw increase in the level of IP3R2, and when we treated with the phenylthyroidea, which is the depigmentary agent, we saw decrease in the levels of IP3R2. And in the mouse model system, Nadal et al. Developed a very beautiful mechanism, in which when we seeded a very low density of cells, that is 100 cells per centimeter square, in the Christian cells will get pigmented from day 0 to day 7 without giving any stimulus. Here we can see clearly here that from day 0 to day 7 the cells get pigmented. And when we check the level of apical isoforms here, of all isoforms, we saw there's a very drastic increase in the level of epithelial 2 in comparison to the epithelial 1 and 3. And then next we characterize all isoforms using siRNA, and we saw there's a specific node of that particular siRNA, and it doesn't affect other isoforms of this epithelial. And in epithelial 2 we saw some kind of compensatory effect in this epithelial isoforms, but we saw there's a clear node of with these siRNAs. And when we checked the LD pellets, and we saw there's a clear decrease in the pigmentation by node in the pigmentation by node of epithelial 2 in comparison to the SINT. And we also validate this using alpha-MSH induced pigmentation and also anti-melanocytes. Yes, ma'am. Okay. So here we saw clear decrease in the pigmentation by node of epithelial 2. We also validated this in vivo, where we node epithelial 2 using morphino based injection at single cell stage in zebra fish, and we found that by node of epithelial 2 we saw decreased pigmentation in jellyfish larva after 48 hours of post fertilization. And we validated this using melanin assay, and we saw decreased pigmentation in epithelial 2 morphino condition. We also did rescue students in the background of epithelial 2 morphino condition, and we saw increase in the level of epithelial 2 by rescuing this epithelial 2, and we saw increase in the pigmentation when we compare with the epithelial 2 morphino condition. By rescuing with epithelial 2, we saw increase in the pigmentation of this jellyfish larva, and we validated this using epithelial 2 using melanin acid, that by rescuing we saw increased in the pigmentation. And next we then checked the mRNA and protein levels of these melanin regulators upon epithelial 2 nodon. Here we saw that by nodon of epithelial 2 we saw drastic decrease in the mRNA level of at 48 hours of post translation, but we didn't see any change in this key melanin genes at mRNA level. And when we checked the protein level, we saw there's a drastic increase in the protein level of these key melanin genes, that is GP100, tyrosine, and district by node of epithelial 2. So here what we found is that the key immunogenic proteins were increasing, but nodon doesn't affect the translation machinery. So how it is increasing, and so to check this, we asked a question that these type of nodon increases the immunogenic proteins half-life. So we used the cyclohexamide change assay, where we but after nodon of FB32, we saw decrease in the level with increasing time of this protein cyclohexamide, but we saw there is an increase in the level of key immunogenic proteins by nodon of FB32. So this gives us that by nodon of FB32, we saw decreased melanoma protein degradation. Even we saw there's a clear decrease in the melanin content. So this is very unusual, but we read literature and we found that whenever cells shows proteasome emission, they automatically activates the autophagy to as a crab protective response in human in cells. So to validate this, we checked autophagy in our system by nodon of epithelial. So first we checked the level of SC32, and we checked and we saw that by nodon of epithelial we saw increased level of SC32. So and we to validate the autophagy flux, we used the vancomycin. So vancomycin actually inhibit the binding of autophagosome to lysosome. So in epithelial nodon concern, we saw accumulation of this nanosome. This autophagosome, and found that there's an increase in the level of SCA2 in compared to the SINT. We can clearly see through these bar graphs. And we also did with this another system using confocal microscopy using GFS3 RFP, in which the GFS3 were get degraded when more autophagy flux is there. So here we can clearly see that from in applied to nodal condition, we saw increased degradation of GFP in compared to the SINT. So this means by nodal of FB32, there is more increased autophagy flux is there. As you saw here that there's a decrease stimulation, so it is more likely that it is because of the nanosome degradation. So we checked the nanophagy. So here we developed a probe named as mCherry Taxonis EGFP. So this probe working as a that GFP when goes into the more acidic environment, that is the lysosome, it actually quenches the process of GFP, but it doesn't quench but it doesn't affect the RFP. So on this concept, we developed this probe, and this here the tyrosine is actually exclusively present on the nanosome. So it only shows the division of the nanosome. So here we can clearly see that by nodon of FB32 we saw increased degradation of this nanosome, and there's increased nanophagy flux. And we also developed another probe using chyma. So chyma is actually a coral protein in which it actually fluorescence red when it goes into the acidic environment, that is lysosome. So we tagged that with chyma with a tyrosine. So it only shows about the nanophagic flux. If the nanosome goes into the lysosome, so it fluorescence red, otherwise not. So here so here we can by the ratiometry we validate this, that by nodon of FB32 there is more nanophagic flux is there. So after this we as autophagy is being going on, there's a change in the pH of the lysosome. So we checked through two different methods, that is one with the lysosome yellow and blue DNT160, and with one with RPH fluorine 2. So here if there is more yellowish color in compared to blue, so it is so more HD condition is there. And when and where there's a 405 by 480 nanometer ratio is less, this means there is a less pH in this system. So by nodon of FB32 we saw there is a less pH in the lysosome. We also checked in the LPDP that is lightly pigmented and pip- uh, darkly pigmented cells, and we found that in the darkly pigmented cells there's more pH in compared to LP, as yellow and blue ratio is down. So after this, we showed that by node now of habitat 2 we saw decreased lysosomal pH. After this, in the as we saw by node now of habitat 2, the habitat 2 actually release calcium from the ER to the lysosome or to mitochondria. So our hypothesis is that by node now of habitat 2 we saw decreased calcium in the lysosome. So on this hypothesis we worked upon, but we found just opposite, that by node now of habitat 2 we saw increased calcium in the habitat node condition. So it is surprising for us that how it is happening. So first we validated this, that by node of habitat 2 we saw increased calcium in the lysosome. We validated this with calcium imaging using diflunisine A1 as a treatment. So it actually releases calcium from the lysosome, and we measured using FeO2 AM probe, and found that in the node of habitat 2 there is more calcium released from the lysosome. We also checked in the LP and DP. Here we saw in the LP condition, that is in the lightly pigmented melanocytes, we saw increased calcium in the lysosome here. So how it is happening? So it is because because of the leaky channels present on the ER. So because of these leaky channels, it is likely that more calcium is being released through this leaky channel into the lysosome by node of habitat 2. So we checked the leaky channel activity by node of habitat 2 using thapsigarmin. Actually, thapsigarmin releases calcium from the leaky channels of the ER. So by putting this thapsigarmin, we saw more calcium is being released through the leaky channels of the ER. So because of this, it is going into lysosome, and it increases the calcium in the lysosome. So after this, the we as calcium is in the lysosome, so it also calcium in the lysosome, it actually releases more calcium to activate autophagy or not. So we checked the level of the activity of TPL1 by node of habitat 2. So here what we found is that by node of habitat 2 we saw increased release of calcium through TPL1 using MLS1, which we particularly release calcium from the TPMLs. So here we used this, and also we used TPL1 Geco. We saw increased calcium release from the node of habitat 2. So in the context of node of habitat 2, we note down in the that both SINT plus SIT1 and SIP plus SIT1. So what we found is that by node of both SIP plus SIT1, we saw decreased calcium release from the from this lysosome. So this means it is in the in the downstream mechanism of habitat 2, that by now this the release of calcium from this triple one. And we also overstress in the background of habitat nodal condition triple wild type and triple mutant. Actually, the triple mutant actually didn't release calcium from the terminals. So what we found is that in the background of habitat 2, we saw a more release of calcium in the triple wild type condition, and in the triple mutant condition, it is almost similar to the pcDNA. So this means that in the downstream of node of habitat 2, there's more calcium release through the triple one. So we checked the nanophagy after knocking those down of these both SINT plus SI1 and SI2 plus SI1 triple one, and we found that there's a less calcium reached through the triple one by Norton of these both. We saw less nanophagy, and when we overexpress this tune wild type and tune mutant in the background of epithelium 2, we saw decreased increased nanophagy here, and there's a no and there's a no nanophagy, or there's a reduced nanophagy in the chromosome of PCDNA, as there is no calcium is being released to the triple mutant here. So this shows that the release of calcium from the triple one is required for the nanophagy in the background of epithelium 2. And we also check the T5 transcription into nucleus, as T5 transcribes many key players of autophagic genes. So here what we found is that T5 actually transcribes in the epithelial nodal condition, and we also checked through using substitute fraction, and we found that T5 actually transcribes into the nucleus more in the epithelial nodal condition. So when T5 transcribes more, it causes more autophagy. So in the summary, what I found is that when alpha is bind with MC1R, it causes release of IP3, and it releases calcium from the ER, and it causes normal basic nanophagy. But in the epithelial Nowton case, what it happened is that when Nowton epithelial 2, it was actually I skipped the this mitochondria part because of the time and the limitation of slides. So here what it happened is that there's a less calcium going into mitochondria, and through leaky channels, it actually calcium is going into the lysosome, and it causes lowers the pH, and more calcium is being released to the triple L1, and it causes dephosphorylation of T5, and T5 then goes into the nucleus, and it transcribes neurogenic genes here, and it starts the alpha initiation complex, and it causes enhanced nanophagy. So through this process, it actually the all this nanophagy pathway is even going on, and in last I acknowledge my supervisor, Dr. Rajendra Motiani, my executive director, Dr. Arvind Sahu, and all my SSV lab members, RCCF and technical staff, CSF for fellowship, and thank you. Open for questions.
Thank you, Suman. Very nice talk, and you finished well within time. And we appreciate that you respected the slide limit.
Okay.
So thank you so much. I'll hand over to Salim, Ashutosh, and our other expert mentors to please, Parveen, to please take some questions and feedback.
I have one my question. So you described mostly about R2. So what do you think R1 and R3 will be doing in this case?
Actually, R1 R3 is actually is not actually taking part in this pigmentation. When we noted this epithelium 2, we saw less release of calcium, like when Norton of epithelium 2, we induced with histamine, and we saw less release of calcium from this from the ear. But yeah, there's a very, we can say there's a very negligible role of this epithelium 1 and 3. It doesn't play any role in this.
And maybe second my question is, so does calcium really change the pH in lysosomes, or do you think there's another pathway involved?
Please repeat the question.
So is it only based on calcium concentration in lysosomes that you think that the pH has been changed, or there are also some other factors or pathways involved?
No, it is not because of the calcium. It is actually because of changing of pH is actually we checked also the ATP6 V1H, also the channel that it is that it is expressed more in this that in the epithelial Norton condition, and also we checked like yeah, that it is that one of these channel we have checked, and yeah, the calcium, calcium is increasing, and it's through leaky channels, and we checked mainly I only checked this channel that 86 V1H is increasing, which is inhibitor to nodal condition.
And can we go back to your first slide, like the introduction slide?
Yeah.
I just have a comment, like nothing special about, like,
Yeah, this one.
yeah, so this one. So for a non-expert, this slide becomes overwhelming sometimes because there are so many things are involved. So if you can show something like stepwise, or when you're talking about this protein, this protein, this protein, you can put some pointer. Like now I'm going to talk about this. Now next this, next this.
Okay.
Like not as the pointer like when you're doing it, but like some animation or something.
Okay.
So if the person who is not really the expert, maybe get a clue where to look at the screen, because now there are so many names on the screen. So sometimes that's overwhelming.
Okay.
Yeah. I think that's it from my side, Krishna.
Yeah,
Sure.
can I come in, Karishma? Yeah. Suman, I thought it was a very nice talk. Although I agree with Pavin, I think you had the data was a bit too overwhelming. The slides were very dense, so that is a general comment I think you should consider for your future presentation. And coming to the work, I had a couple of questions. So you showed a very interesting thing somewhere in the middle where you talk about the melanosome lysosome fusion.
Yeah.
Okay, for the knockdown. Can you go back to that slide? I think it was imaging data.
This one?
No, not this one. So this was about the knockdown that is basically enhancing the rate of melanosome lysosome fusion.
No, lysosome calcium actually. It actually by knockdown of FibThR2, there's a more increase in the lysosomal calcium. Lysosomal calcium it is.
No, there was a schematic in one of your slides.
Yeah, it is this one.
No, not this one. Go back. It's a cartoon that you showed.
Yeah, this one?
Yeah, here.
Okay.
So this is what I was, So basically in your knockdown, your melanosomes and lysosomes fuse, right? That is what you're implying?
No. It is the melanophagy in which the melanosome is fused with the lysosome for the melanophagy.
Yeah. So what are the sizes of these melanosomes in general? Like how large are these?
It is around similar of the it is actually the lysosome related organelles. So it is the actual size I didn't know about what the actual size is there.
Like
But
roughly, I mean, are these like below the diffraction limit, or are these larger than 300 nanometer, 500 nanometer, or smaller than that?
I didn't know actually.
Okay. So the reason I'm asking is because since you're showing imaging data, and if we are dealing with, let's say, any vesicles that is much smaller than the diffraction limit of, let's say, 250 nanometer, then we should be cautious in concluding that they're actually fusing. They can be sitting close to each other or in a crowded environment, but not really be fusing.
Okay.
So unless you like double check that with super resolution, you might not really know whether they are actually fusing or whether they are just sitting next to each other, because that would also show you the same co-localization signal.
It's not the co-localization, it is a lysosomal degradation, like the mitophagy or eophagy or any phagy. It is like the melanosome degradation through autophagy. It is a organ dependent sensitive to organ, that is it is the melanosome degradation, that is we call it the melanophagy.
Okay. But there is some fusion happening, right?
Fusion, we didn't show about the we didn't talk about the collocation here. We are talking about the nanosome degradation here.
Okay.
That nanosome get degraded by like autophagosome fused with the lysosome, so it get degraded. So it is in the same way nanosome get fused with the lysosome and it get degraded in the
Yeah.
same way. And nanosome is the it's a lysosome related organelle, so it is almost of the similar size to the lysosome, because it's a lysosome related organelles. And most of the proteins were also present on the nanosome. AC3 is also present on the nanosome, and some kind of like TPC is also present on the nanosome. So it is like a very similar organelle of lysosome, but it has some different different proteins like tyrosine, DHT. Like these are proteins which are in the pigmentation. So as pigmentation is going down, so we checked that that particular organelle is getting degraded. As we saw that these proteins, melanin proteins, like in normal cases, what happened is that when pigmentation is going down, we saw a decrease in the melanin proteins here. But in my case, what it is happening that by knockdown of epithelial 2, pigmentation is going down, but we saw increase in the key melanin proteins. So first we checked the autophagy, and then we move it to particular to the nanosome degradation, and we found that nanosome get degraded by knockdown of epithelial 2, and because of this nanosome degradation, we saw decrease in the pigmentary phenotype.
Okay. Okay. Yeah. And the other question I had was, so you mentioned about some leaky channels. So are there any particular leaky channels?
Like we didn't check what the particularly leakage channels, but through topsy-gargin here, actually topsy-gargin releases calcium to the leaky channels. So here, what it is found that by knockdown of the leaky channels, here it actually the activity of the leaky channels is actually increased by knockdown of epithelial 2, and this causes more calcium released through these leaky channels.
Right.
And we also checked that by knockdown of epithelial 2, there's more accumulation of calcium in the ER. I didn't show that data, but it is also I saw that by knockdown of epithelial 2, there more calcium in the ER, and the leaky channels are more active, so more calcium is being released through the ER. And this calcium is going to the lysosome as here epithelial 2 is knocked down. So as in if we saw the previous literature, there's a sole channel for driving calcium to the ER to the lysosome or mitochondria is the epithelial. But here we knocked down epithelial, and we still show increase in the lysosome here. So it is very much surprising for us also.
Right. So maybe it would be good to, like, in future try to look into which leaky channels are responsible. Yeah. I think I'm that's it from me.
Thank you, Salim. Thank you. All right. Ashutosh, we
Yeah.
have a couple of minutes left. Go
Yeah.
ahead. Go ahead.
Yeah, so very nice talk, Suman.
Thank you,
Quite
sir.
a lot of work we have presented here. I completely agree with what Parveen and Salim have actually said regarding the overwhelming amount of data, especially for people who are not from the field, let's say, exactly from the field. Also, the other thing is that in the first introduction slide, when you are showing, it may be better to just focus on the areas that you are going to talk about in the rest of the topics. Okay. And because people have very short memories, including my own, it helps to actually keep reminding them. So you can put this slide of schematic in between, that now I'm talking about this and now I'm talking about this. It just keeps reminding people that, okay, you're talking about now this step of the work. Okay, that's a very general kind of comment. Just one question. From all of this work, what have you understood from all your study which was not known before? If you could summarize it in one line, what would that be?
We can say that it is the role of epithelial in nanophagy. It is actually didn't know before that, and also the the calcium actually with the burnout of epithelial 2, the calcium increase in the lysosome is increasing. Burnout of epithelial 2 is actually new in this field or in any we can say, because not only the in the skin biology, but it also in all the fields. It is very much new that burnout of epithelial 2, it is the calcium is increasing in the lysosome, and it causes through tryptophan it causes autophagy, or we can say nanophagy is there.
Great. Okay. Thank you so much. Going forward, you may want to think along the lines of from the findings that you have now, is there a translational application that you can think of it, or clinical relevance of this that you can keep in mind? I'm sure that you've thought about it. We don't have time to go into that, but
Okay.
it's nice to highlight that.
Thank you.
Perhaps towards the end of the talk. Okay.
Thank you. Thank you.
That's very nice work.
Thank you.
Thank you to all the three of you, and thank you, Suman. Well done.
Yeah, thank
Best
you.
wishes in your onward professional journey. And yeah, thanks for your interest in PhD Cafe. You can stop slide sharing now, Suman.
Okay. Okay.
And our next speaker is Kinjal. Kinjal, I see you are ready. Welcome, all the best. And Kinjal is going to talk about snares in membrane fusion.
It is
Snares being an acronym. So Kinjal, you can start screen sharing now.
actually from where it is to stop.
Usually, Suman, at the bottom panel, it says stop share. There'll be red and green buttons, and the red will say stop share.
Yeah, I found.
Yeah, got it.
Thank you.
Great, Kinjal. You can start slide share.
Okay. Are my slides visible and am I audible?
Yes.
Okay.
All the
So,
best. 20 minutes, Kinjal. 20 minutes.
Yes. Okay. So, good morning, everyone. I'm Kinjal from Tata Institute of Fundamental Research. And as correctly pointed out, I work on understanding the structure-function relationship of snares and membrane fusion. To begin with, membrane fusion plays an important role in just virus entry to the host cells, vacuole fusion inside the yeast. Even in the eukaryotes, the intracellular vesicular pathways as well as cell-to-cell communication pathways.
Now we can. Yeah.
Okay. So membrane fusion is an important process in various phenomena such as viral fusion to the host cell, vacuole fusion inside the yeast cells. Even inside the eukaryotes, the vesicular transport mechanisms in the intracellular vesicular transport mechanisms, as well as cell-to-cell communication pathways, such as synaptic vesicles fusing to the plasma membrane, undergo membrane fusion events. Now, membrane fusion is a highly energetic process. Therefore, there's a need of a mechanism to overcome this energy barrier to catalyze this entire process. And this process is catalyzed by a family of proteins called as snare proteins. In brief, snare proteins are anchored to two different sets of membrane, undergo sequential hydrophobic interactions, bridging the gap between the two membranes and catalyzing the process of membrane fusion. For my entire talk, I'll be focused on snare proteins which are involved in fusion of synaptic vesicles at the plasma membrane in the presynaptic terminal, mainly synaptobrevin 2, which is present on the synaptic vesicle, also called as V-snare, or T-snare proteins, syntaxin and SNAP25, which is present on the target membrane, in this case, the plasma membrane. Now, if we go into the details of brief events of which occur during the synaptic vesicle fusing to the plasma membrane, it undergoes various steps of synaptic vesicle being held close to the plasma membrane, undergoing fusion and then the recycling. For this talk, I'll be focused on the key steps of docking, priming, and then fusion. So, synaptic vesicles are held close to the plasma membrane, and this step is regulated by calcium sensors called as synaptotagmin 1. Synaptotagmin 1 binds to the snare complex and arrests it at a half zippered state or a semi-coiled coil formation. And when an action potential is received at the nerve terminal, there's an influx of calcium due to voltage gated challenge, resulting in the formation of a fusion pore, which is the first aqueous connection between the intracellular of the intravesicular lumen and the cell exterior. Thus, this excitation secretion coupling holds the key to when an action potential is received and the signal is then transmitted. Now, if we go into the release events which happen during these that there are different types of releases, such as the one that I spoke now about is the synchronous release, which occurs when there's an influx of action potential is received and there's an influx of calcium molecules. And this is majorly regulated by a calcium sensor called a synaptotagmin 1. But there are different other types of releases which also occur at the synapse, which is asynchronous release, which is characterized, which is regulated by synaptotagmin 7, another isoform of the synaptotagmin family. Also, there are spontaneous fusion events which occur, which is regulated by DOCK2 protein. Surprisingly, all these proteins work at different calcium concentrations within the synapse, but all of them work via the same snare machinery. So our key question is how these different calcium sensors, which have the downstream different fusion events, regulate via the same machinery of the minimal snare machinery. To then deal with, so this leads to understanding why understanding the structure function of this snare minimal machinery is important. When we go into deep into the domains of a snare, synaptobrevin 2, which is the protein present on a synaptic vesicle, has 3 different domains. The first snare domain undergoes sequential hydrophobic interactions from N terminus to the C terminus, thus catalyzing the process, releasing energy and catalyzing the process of membrane fusion. The transmembrane domain also is not just a mere lipid anchor, as when we in literature, when people replaced this transmembrane domain with certain lipid anchors, it failed to support exocytosis. Now, surprisingly, all the various regulatory proteins bind at this snare motif. However, the pore properties are governed by this transmembrane domain region. So we asked a simple question, is there an allosteric or functional coupling between these two regions by which it can facilitate the regulation by different regulators? The simple methodology is to understand the linkage between snare domain and transmembrane domain, we introduced site-direct specific mutations at these residues, purified these mutated proteins in vitro, incorporated them into proteolipidic entities, one being nanodisk, which is the flat lipid bilayer surrounded by a membrane scaffolding disk, the membrane scaffolding protein MSP, restricting its diameter to 13 nanometer, and to which the synaptobrevin 2 or SIP2 proteins are incorporated. And there's a proteolipidic liposomes, which is nothing but a spherical lipid bilayered vesicle, to which we incorporate the T-snare proteins, which is syntaxin and SNAP25. And we mix them together to perform our in vitro fusion experiments. To start with, I will start with a simple in vitro experiment, which is called as lipid mixing experiment. In brief, the labeled nanodiscs contain labeled FRET pairs, NBD and rhodamine labeled phospholipids, and this is mixed with unlabeled liposomes containing T snares called as T liposomes. When we mix them together, it will initiate membrane fusion reactions, resulting in the mixing of lipids in both these entities, as a result of which this FRET pair is now separated, and you can record the donor dequenching or the NBD fluorescence with time, which gives you a proxy of the membrane fusion events. Now we used these experiments with various single and double mutations. Most of these, we have 3 mutations, M46A, R56, and F77. These 3 sites are on the snare motif, which is mutated for this study. And there's another mutation is L99, which is on the transmembrane domain. So all the pairs that I have so far is one from the snare motif and one from the TMD. So the first pair that I have is M46A and L99W. Both these single mutations show reduction in the maximum of this lipid mixing percentages. And when we compared the time kinetics or the half time, which is half time is the time at which the half maximum is reached, we observe that both of these mutations, M46 and L99W, is comparable to half time. However, the double mutant M46A L99W has a significant reduction in the maximum percentage, as well as the half time increases significantly as compared to wild type. We move on to the next pair of R56P and L99W. And we observe that all of these mutations, the single and the double mutations, are very comparable to the wild type. Even the half times are comparable to wild type. The third pair that we compared so far is F77S and L99W. This also shows a phenotype similar to the first pair, that the double mutant is significantly the maximum of double mutant is very low, as well as the time kinetics of half time of the F77S L99W is significantly higher as compared to the wild. Now, so far we just observe that some of these mutations show an impact on the membrane fusion events. We wanted to gain deeper insights into whether these are coupled or not. We implied the double mutant cycle analysis. In brief, double mutant cycle analysis is that there's a protein P which is wild type, and you have two proteins mutated, PS where the snare motif is mutated and PT where the transmembrane region is mutated. Now you have a double mutant where PST, both a snare motif and a TMD motif is mutated. Now if PST is coupled, if the residues PS and PT are coupled together, then the energetics of these PST values are significantly or synergistically higher, and it will be non-additive. So as stated in this formula over here, that if the delta delta G values of ST is higher than the additives of S and T, it indicates coupling between these residues. So we calculated the delta delta G values for each of these pairs, and we observed that the first pair M46A and L99W to be synergistically higher than the additives of this, indicating this is a positive coupling. In case of R56P L99W, we don't see such an increase in the double mutant, indicating it is not coupled. The third pair that we tested here, although there's an increase in the half time values, the delta delta G values is additive of the F77 and SL99W, indicates that this is also not coupled. So we observed one pair to show allosteric coupling. We move on to another experiment set up in our lab, which is called as NDBLMS experiment. In brief, there's a cis and a trans chamber separated by a small aperture. On this aperture, we paint lipids and add T-snare proteins or the target snare proteins, syntaxin and SNAP25. To the cis chamber, we add nanodisk containing V snare proteins, and when it undergoes fusion, there's a flux through through this, and you can record current versus time. So basically, we can record single pore and at sub-millisecond timescales and observe whether these mutations alter the pore properties. To begin with, there's a wild type protein which shows mostly stable open pore with very few fluctuations towards the closed state. When we compare the single to single mutations, it also shows very stable pore phenotype, fusion pore phenotype. However, the double mutation shows a lot of flickery behavior and transitions from closed to open state. From these raw current versus time graphs, we can plot conductance values, which is a proxy for the size of the fusion pore. And we observe that M46A and L99W shows a higher conductance values. We can also plot the fraction closed. It's basically the time spent in either the closed state or the open state. And as observed from the raw traces, we observe the fraction closed values of M46A and L99W significantly higher, indicating the instability in the fusion pore. We also plot the open time spent in the open dwell time. This will basically give us a proxy for stability, and also from here we can calculate the rate of this reaction. When we fit this open dwell time, CDF of this open dwell time, it's a 2 exponential fit, and we take the slower rate, we take the time constant of the slower component to calculate the rate of this reaction. And we observe that again implying the double mutant cycle analysis, we imply that this double mutant has significantly higher delta delta G values as compared to the single mutations. We move to the next pair, which is F77S and L99W. This again shows a similar phenotype, that the double mutant has more flickery behavior as compared to wild type. Although it does not show any changes in the pore size or the conductance values are comparable to wild type. The fraction closed shows that a higher fraction closed values for F77S L99W, indicating that this pore is very flickery in nature. Also, when we plot the F77S CDF values of open dwell times, we observe a sharp shift towards shorter open lifetime of F77S L99W. And this is also indicated in the delta delta G calculations, where the delta delta G values for F77S L99W significantly higher, synergistically higher. So this is interesting because this mutation F77S and L99W was previously found to be additive, and here it is synergistically higher, indicating that there may be certain uh, allosteric pairs which act at different stages during the entire vesicle fusion cycle. Now, why at all this is important to study is that, coming back to the major question in our hypothesis, is that there are different calcium sensors which act at different stages, which act by the same snare machinery and characterize different types of fusion events. So to begin with, we start with one of the calcium sensor, which is synaptotagmin 1 or SIT1, which has a transmembrane domain region and two major calcium sensing domains, C2A and C2B. For our study, we use just the C2AB region, the calcium sensing regions of these two proteins. And we established the role of this synaptotagmin 1 in this NDBLM setup that we have over here. So in the wild type pore, when after the pore is open, we add various concentrations of synaptotagmin 1 in the absence of calcium. And we observe that at 7.5 micromolar of the calcium sensor, synaptotagmin 1 C2AB, the pore closes completely, suggesting this phenocopies the physiological role of synaptotagmin 1, that in absence of calcium, it does not allow fusion events. Now to this closed pore, when we add titrate calcium concentrations, we observe that at 500 micromolar of calcium, the pore again opens up. This is also phenocopies the physiological role of synaptotagmin 1, that when the calcium influx is received, the clamp of this synaptotagmin 1 is relieved, and there's an opening of the fusion pore. This changes in the open and the closed state, it was also denoted by the current changes, where you, on addition of synaptotagmin, the current shifts towards mostly closed state, which is at 0. And at addition of calcium sensors, it again shifts to an open state, which is somewhere around -10 to -20. This is the titration curve that we observed by using different calcium concentrations and the set, sorry, and the regulator, and we observe that this entire curve saturates at roughly around 7.5 micromolar of synaptotagmin 1, and the calcium titration saturate nearly around 500 micromolar. So we use these final concentrations and then now combine our mutation analysis study with this calcium sensor studies to study how this calcium regulation occurs in presence of different mutations. So the first pair that I've tested so far is the M46A and L99W, reiterating that M46 is on the snare motif and L99W is at the transmembrane domain. And we observed that in the presence of 7.5 micromolar of SIT1, the pore closes in both the wild type as well as the M46A L99W, as also indicated in the fraction closed values, where you have higher further increase in the fraction closed values upon addition of SIT1. However, we could not go into the further kinetic analysis of this pore, as we need still more repetitions to complete the story. Also, we need to more focus on what happens to this closed pore when you add a calcium concentration. So far, I could only reach the 5 micromolar concentrations, and so far it looks good that the pore remains closed. We move to the next mutation, which is F77S L99W. Surprisingly, what we observe over here is that although spore closes at 7.5 micromolar of synaptotagmin 1, it opens at very less calcium concentrations of 5 micromolar. The pore again opens up. When we compare the fraction closed values for these proteins, we observe that in the presence of wild type, there's a significant increase in the fraction closed upon CYT1 addition without any calcium. But in the presence of CYT1, there's no difference in the fraction closed values in the F77S L99W mutation. Now, this is surprisingly interesting because synaptotagmin 1 binds to the snare motif somewhere at the 0th layer, which is just before the R56 values. But the mutations that we've observed is downstream of CYT1's action, and yet mutations at these residues affect the CYT1's regulation. We also observe a shift in the open dwell time. Initially, the open dwell time of F77 SL19W is mostly shorter open times or mostly closed. However, that is shifted towards the right with the synaptotagmin addition. We further need to repeat these experiments to consolidate our results and go into the energetics of this. So summarizing the work that so far, we have observed that there's a functional coupling within the snare motif and the transmembrane motif of the synaptobrevin 2. Synaptotagmin CYT1, which is just the functional region of synaptotagmin, can act as a fusion clamp in the absence of calcium. So this itself is enough to characterize the functional regulation of the synaptotagmin 1. The synaptotagmin 1 mediated regulation is abrogated in the presence of synaptotagmin F77S L99W mutation, indicating the importance of this coupling mechanism. We need to perform further experiments, perform MD simulations to understand the structural variations in the synaptobrevin 2, because of these mutations, which might help in understanding how this regulation might be affected. And we also intend to perform these similar experiments with different calcium sensors such as DOC2B to understand whether the regulation act via the same allosteric links or it is different for all the different calcium sensors. Lastly, I would like to thank Debashish, my supervisor, to give me the opportunity to study this entire work, my all lab members who have been helping me through for these studies, our department, TIFRDAE for funding, and the expert mentors and India Bioscience for giving me this opportunity to present my work. Thank you.
Well done. Kept to time as well. Now we'll get some questions and suggestions for you from the expert mentors.
Yes.
Yeah, so maybe I can go ahead, if that's fine.
Sorry, you said something, Ashutosh?
Can I go ahead?
Yeah, sure.
Right. Yeah, very nice work, Kinjal. Very detailed, very nicely presented. I mean, the presentation was all very good. I felt that some of the figures were a little small. They can be made bigger, but that's more of a personal choice kind of a thing. So I have a couple of questions with regards to the mutations that you studied. How did you identify those mutations? Perhaps I missed that in the initial part, but what was the basis for those?
So just a brief understanding is that the entire snare motif is like 15 hydrophobic interactions. So we plan to screen all of them sequentially. But for the preliminary study, what we have selected is there is a 0 central layer, which is R56, and there's one towards the N terminus and one towards the C terminus. So it's randomly selected just to span the entire snare motif. Snare domain. We plan to sequentially increase. So if we observe something towards the C terminus, we'll scan few residues towards that end of the protein. Or if we get something towards the N terminus, we'll scan that part. This was the preliminary idea.
Okay, and you mentioned that there is a coupling that is there in M46 and L99, if I'm not wrong, right?
Yes.
Can you identify that in the figure, please? Where?
This is M46, which is here, the red spheres over here, and L99W is on the lipid bilayer, which this is a on the transmembrane domain, which is the.
So, what do you think, in your opinion, is the mechanism for coupling that exists here?
So there are few hypotheses that I have in my mind. So this entire protein is a helical spring sort of a thing. So introducing mutations can alter either the phase of where so this transmembrane domain is where the core properties lie. So if you have a mutation over here, it can introduce rotational conformational changes at the transmembrane domain, which these residues might not face at the correct phase of the fusion pore, or it might hamper the insertion of these proteins at the membrane fusion. So what I see is it's just a energy transfer mechanism. So introducing two simultaneous mutations, the energy which is transferred to the membrane, which is required for the catalysis of the membrane fusion, might be hampered because of these sequential mutations, which might lead to the effects that we see so far.
So you mentioned that there is a catalytic mechanism. What is that catalysis that we are talking about here? Sorry, I'm not aware with the system. So
Yeah, no problem. So if we just go into this detail, there's a gap between the two membranes. So it will be it's like two hydrophobic residues separated by an aqueous cytoplasmic domain. So
yeah.
you need to bridge the gap between the two membranes to less than 2 nanometers, such that it can now spontaneously fuse. Now this gap is catalyzed by the process called a snare protein. So basically these are just intrinsically disordered proteins. What happens is that when it encounters another snare pair, which is the present on the which is present on the plasma membrane. What happens? It undergoes this coiled coil domain formation, which is nothing but coiled coil zippering events from the N terminus to the C terminus. What happens? It then pushes the two membranes closer and closer enough to catalyze the entire process.
Correct. I was just wondering where is the energy for this coming from?
Energy is coming from these hydrophobic interactions. So it undergoes 15 hydrophobic interactions sequentially. And when you, it's basically forming hydrophobic interaction, releasing energy.
It's primarily enthalpy driven in that case.
Yes.
Okay. All right. All right. I have no further questions. Thank you so much, Kinjal. It was very nice presentation. Over to you, Parishma.
Yeah, Salim, Parveen, either of you. Yeah.
Yeah, hi Kinjal. Very nice work. And say my hi to Devashish, by the way. Yeah.
Yes.
So I had a couple of questions. So I'm wondering, why did you go for your fret assays? Why did you go for liposome and a nanodisk and not two liposomes?
Okay, interesting
Is there
questions.
any particular reason behind that? Because nanodiscs, I'm sure you are also aware there are certain caveats there, right? So
yes.
Yeah.
So our primary goal was to understand our primary goal was to understand the fusion pore. Now, when we use two different liposomes, what happens is you can't arrest it at a fusion pore. So it basically entire two liposomes will fuse together to form a larger liposomes. The advantage of using nanodisk is that the diameter is restricted to 13 nanometer because of these membrane scaffolding proteins. So when the nanodisk fuses to the liposome, it arrests it at a half. It arrests it at a snare complex, or arrests it at a fusion pore state, and does not allow the full dilation of the fusion. And that is the reason we chose a nanodisk and liposome system.
Right. But do you think, since you're restricting the size of the nanodisk, the curvature aspect, as well as the, let's say, the lateral pressure or the tension might, get modulated, and that might also affect the dynamics of your the snares?
Yes, it will. So we are planning to do. So you can modulate the size of the nanodisk based on what MSP protein you use, and we have certain larger nanodisks of 50 nanometer in diameter in our lab, and we plan to do these experiments with those larger nanodisks as well to find out whether the restricted diameter or the curvature is playing a role in modulating these, because as mentioned, snares alone don't act. It's also the lipid environment which acts together.
Exactly.
For this.
Yes. Yeah. Right. And do you also, I looked at the composition that you're using. Are you planning to also look into some lysolipids?
Not not currently. We plan to maybe incorporate cholesterol because cholesterol is also a major constituent of synaptic vesicle protein.
Right, right. So I mean, it would be interesting also to look at, since you were like looking at the docking and the formation of hemifusion. So if you can, I mean, you might think about it and having some lysolipids, especially at the high curvature region, might also be in the liposome at least. May not be in the nanodisk, but that would also be interesting to look at because of the geometry, the head to tail aspect ratio of the lipid. So you might think about it. Yeah.
Yeah, sure. Thank you.
Okay, very good.
Thank you.
Yeah. So actually, I had exactly same questions with Salim and Ashutosh already asked, and very nice presentation, by the way. So coming back to nanodisk, so it is not entirely true that your nanodisk size remain the same. I have one published structure now, and then there is another one which I was writing the manuscript. Same nanodisk form different sides with nanoproteins. So you have to be very careful interpreting nanodisk data because not that many structures or protein structures are available. So they can still modulate a little bit and they can take slightly different shapes when you put your transmembrane domains or membrane proteins in those ones. So yeah, I think it would be nice if you can try these experiments with the different size nanodisk. So that doesn't make any difference. So like in my case, what I've seen with the membrane protein, one of the membrane protein is when I changed the nanodisk, there was change in the helical arrangement. So we are still debating whether we should put in the manuscript saying it's the effect of nanodisk or it's an alternate conformation. So nanodisk can also give you some artifacts. So you have to be very careful interpreting these results. And as I said, maybe try different sites, MSP1, MSP2, and like all these N1, N2, and those higher, bigger size nanodisk. So that's very nice question. You said 500 micromolar calcium was able to work for. Is it close to physiological concentration in these systems?
No, it is much higher. So physiological calcium concentrations of this calcium sensor is roughly close to 100 micromolars. But one thing to note is that the synaptotagmin 1, which is in a physiological condition, has a transmembrane domain. It is located on the synaptic vesicle. There are 15 copies of synaptotagmin 1 on the synaptic vesicle. Here we add much higher concentration of synaptotagmin 1 also. So we speculate that because there's a lot more calcium sensors in the solution itself, you will require much higher calcium concentrations to saturate the entire, because each C2AB domain can bind to up to 5 calcium molecules. So that is the stoichiometry of these.
Okay. Yeah. Thank you. Good luck.
Thank you.
Thank you all. That's some really detailed feedback. Really appreciate it, all three of you. Thank you. And well done, Wendell Kinjal. Really nice talk. And our third and final speaker for membrane biology is Avinash Gautam. So Avinash, I see you. You can, yes, I see you've unmuted. Now you can slide share. Avinash, you can slide share, please. Perfect. I think it's showing up. Yes, we can see. You can go into slide share mode, Avinash. And you have 20 minutes. Okay, time starts now. All the best. Avinash, we cannot hear you. You are unmuted and we can see you, but we cannot hear you. Can you say something, Avinash, maybe? No, we can't hear you. Can anyone else hear him? No,
No.
no?
He's not
No, okay.
audible.
No,
Right,
not
right,
audible.
right. Avinash, see, you're unmuted. No, we can't hear you. What could you do? Could you increase, I don't know, try and increase the volume of your microphone?
Maybe change the microphone. So that sometime laptop also takes some other microphone or headset which is connected to
Exactly.
Bluetooth.
Good point, good point. Yeah, exactly. Avinash, can you check if what your system is connected to?
Am I audible? Hello.
Yes, now you are. Now you are. Great. Go ahead. All the best. Yeah.
So it's screen is audible for you.
Yes, yes. Why don't you go to slide share mode? Avinash, why don't you start your talk?
Okay, it's slide is showing like you can see my slide because
No, we can see the slide, but it's like not in slide share. You can see that left panel. And
Yeah.
yeah, now it is in slide share. That's right. Go ahead. Go ahead.
Good morning, everyone. I'm Avinash from School of Life Science, Jawaharlal Nehru University. I am doing PhD under the supervision of Professor Samudra Govinath, and our lab is focused on the biophysics and structural biology. And today I will discuss about my PhD work. Is focused on the unveiling the cytoskeleton mechanics of an time by to freeze its movement to protect us. Ma'am, how can we do the pointer like?
You should be able to just right click and find it. Just right click.
And now go to your slideshow mode, right click.
Slideshow. Slideshow is bottom. Yes.
Yeah, right
Yes.
click and then pointer options maybe.
Okay.
Thank you.
Let's start some beginning introduction. Like amoeba, so the false feet, like walk without leg. So it's called pseudopodia. Pseudopodia are temporary projection of all cell. It is typically used for locomotion and for phagocytosis. So this mechanism used for amoeba. So it is known as also known as amoeboid movement. And amoeboid movement, there are three type of movement: gliding, walking, and swimming. So in this moment, basically, the amoeba has so the construction and extension of the cytoplasmic body. So this for if we see the basic mechanism of this environment is depend on the cytoskeleton protein. So there are two form of mobility, like bleb driven and actin driven mobility. So it is based on the cytoskeleton proteins like actin, myosin proteins. So why we discuss about this amoeboid movement? Because it is involved in very fundamental biological process such as immune response, cellular migration, wound healing, cancer metastasis, and it is very crucial step for the protozoan pathogen for their invasion and infection survival, the pathogenesis. So let's stress about the evolution from human back to human to the amoeba. So in human, there are many immune cells which use the so the amoeboid movement like neutrophil, eosinophil, basophil, monocytes, macrophage, and natural cells and B T cells. So you just show the amoeboid movement. And some lower organism like yeast, dictyostelium have a complex study about this amoeboid movement. And in protozoan pathogen, the Plasmodium, Giardia, Leishmania, Naegleria, it have very complicated cytoskeleton arrangement due to their limited number of cytoskeleton assembly. So for the understanding of this amoeboid movement, it's linked about this pathogenicity, and we can stop their amoeboid movement or pathogenicity by the stop of invasion and survival process. So the pathogenesis of this amoeba is based on their mortality, fast mortality, and phagocytosis process.
So this fast motility and phagocytosis is controlled by the cytoskeleton, actin cytoskeleton proteins. So there are mechanism which that cytoskeleton protein involved, like actomyosin contractility and actin polymerization. So from some modern algorithm like diptylstelium they have a study about this Rho GTP, Rho phosphatidylinositol pathway and calcium pathway. So from there we can explore their pathway in amoeba also. As we understood that the cytoskeleton protein involved in cell movement and phagocytosis, micropinocytosis, leukocytosis. So there are many family of protein which involved to the modulate of the cytoskeleton protein. Earlier, our lab has studied about actin binding proteins, motor proteins, kinases, and phosphatases. And now my focus, my work on PhD in the focus of pro family disease and its regulators, and it is also regulated by 3 proteins, JEF, GAP, and JDI proteins. So this is the schematic representation of pro-JEF GTPase signaling pathway. In all organisms, there are some conventional pathway like how it play the signaling in the organisms. So this is the conventional pathway where PIP3 to the actin proteins. So PIP3 interact with the ROGF and then activate GTPase protein and then activate PAC, and then PAC interact with myosin, and then myosin actin contractility and actin polymerization help to the movement of cells. So we have selected protein from the on basis of literature and earlier our lab work. So this protein in bypass the conventional pathway to transfer the signal to the actin or myosin proteins. So the first set of experiment where we see that myosin directly interact with EHFP10 protein without bypassing the conventional method. So myosin SH3 domain. So this is the structure of model structure of myosin proteins, where this is the head region and neck region and C-terminal region. So the C-terminal region has 3 domain: TH1, TH2, and SH3 domain. So this SH3 domain interact with the C-terminal of EHFP10 protein, and we co-crystallize of SH3 domain and the peptide of C-terminal domain of EHFP10 protein. And also validated this interaction in the cell biology experiment, where we localize the EHFP10 protein and the phagocytosis formation involved in this process. So here we see that EHFP10 involved in the phagocytosis formation, and the myosin also present in the cytoskeleton rearrangement process. So from the signaling protein, this mass spec data where overexpressed of myosin 1B in phagosome. In this data, we saw that EHFP10 protein came with the along with RAGZ protein. RAGZ is a Rho GTPase protein, and PAC is a P21 activating kinase protein. So from here we have taken the idea like ROGF also activated GTPase protein and enter with PAC protein also. Then for the a structural study and for the study of EHFP10, we clone and purify and the structure determination of EHFP10. So the EHFP10 is like 5 domain containing protein. It is a very long 876 amino acid multi-domain protein, and the row it contains ROGF, DH domain, and PH domain, and 5 domain, and C-terminal domain. So ROGF is a catalytic domain protein. It exchanges the GDP to GTP, and PH and 5 supposed to interact with the lipid membrane lipid. And C-terminal domain, you can see the see that C-terminal domain interact with SH3 domain. So here the first we clone for the structural study, we clone it domain wise because the full length protein is unable to overexpress in bacterial cell line. So we clone in domain wise DS domain and DSPS domain. And then we purify by nickel affinity chromatography and followed by the size chromatography. And then we got crystals of this protein. We crystallize this protein, and then we solve the structure and the DS domain contains 7 alpha helices, and PS domain have DS domain and pH contain beta sheet. There are some loop region. The pH and TH is like pH and DH is like flexible type of structure where the row GTPs get bind and then do their activity. And then further, we did the pH lipid interaction study using lipid dot blotter study with using lipid strips. So we take lipid strip and we put there our protein, and then we did the Western blot process where we got that PIP2 and P3 phosphate spot there. So we from here we can assume that the PIP3 PS may be interact with PIP2 lipids. Then we have selected 8 row GTPase protein on the basis of literature. This 8 row GTPase protein involved in the phagocytosis process. So different GTPase protein involve different time of phagocytosis. It is not mentioned exactly which involved in which time of the process, like row 1, row 2, row 4, row 5, and row 6, 8, 13, 15. This 8 GTPase protein involved in the phagocytosis process. So we check about we want to check that which GTPase protein activated by this GTPase AHF pattern Jeff proteins. So from their activity assay, we did the fluorescence labeled man GTP level activity assay, where we incubate the row GTPase protein with manned fluorescence level man GDP. And after incubation, we pass the GTP with in the presence of Jeff and without Jeff. So in the presence of Jeff, it in the presence of Jeff, it exchanges the GDP to GTP, and then we measure the release amount of dissociation amount of the man GDP in the fluorescence spectrometry, where if we got the signal in the presence of Jeff at high concentration, is a high signal. So it means all GTPase protein have intrinsic activity. So, but in the presence of Jeff, it shows the higher activity, the higher dissociation, like high exchange process. So after validate or the interaction, we did the BLI, biolayer interferometry and interferometry. So in BLI, we see the interaction of the protein of HFP10 DHPS domain with RAGG or RO2 proteins. So where we find that first we mobilize the DHPS domain with the biosensor and then pass the different concentration of RAGG protein, and it showed that association and dissociation. So it means the interaction of Jeff DHPS domain with GTPS proteins. So we did this experiment with other row 1, row 13, and row 6, and we got some affinity specificity, like row 2 and row 6 have give some similar kind of affinity, but row 1 and row 13 have less affinity. So it means the this EHFP and DHPS is have higher affinity toward the row 2 and row 6 compared to the row 1 and row 13. And this is the complex model generated from the AlphaFold, where the DH and PH domain, and here the GTPS protein. So here the DH-PH domain is movable, like it facilitates the activity when PH domain is present. Then the third interaction, I want to show that it is also bypass the conventional method, because it the RAGG direct interact with myosin instead of PIAC protein. But we direct interact with myosin protein. Okay. So first we purify the RAGG protein and SS3 protein, and followed by the size spinal chromatography, and then did the BLI, biolayer techniques. So where we first immobilize the SS3 protein in the biosensor and then pass different concentration of RAG protein, and it shows the association dissociation. The KD value is the dissociation constant will value is like 24 nanomolar, and then we aided their interaction by docking study, and in docking it shows like 14 nanomolar, so it means the interaction will be very significant interaction. And this is the interfacing residue between RAG and SS3 domain, and where you can see the residue interact with RAG with this is the residue and this is SS3. So also we perform this experiment with row 1 and row 6, and it had similar kind of affinity, lower affinity compared to the RAG. Then the fourth in I want to show the interaction between PAC with JTPase protein. So in amoeba, there are 7 PAC proteins. PAC is a P21 activating kinase protein, and there is 21 known JTPase proteins. So the first we identify that HFP10 interact with RO2, that is RAG protein, and then I want to find that which the RAG interact with which PAC protein. So on the basis of bioinformatics study, we have selected the PAC4 protein, and then PAC4 protein. This is the alpha fold of PAC4 protein, where at the C-terminal region they are very long disordered region. So for the structural study, we want to crystallize it, and we got crystal, but the crystal is defective to 5 angstrom, and then it is unable to solve the structure. So I'm still trying to solve the structure and improve the crystal quality. And then we did the superimposition of PAC alpha-fold model of PAC4 with human PAC1, and where we got that the same inhibitor occupied a different pocket area. So if it's look like similar, but there are some space where the inhibitor is not fitting as in human also, as in human. So PAC4 have different pocket area where the same inhibitor get fit there. So then we did the BLI with JTPase, RAG with PAC4 protein, and here we immobilize the PAC protein in the biosensor and pass different concentration of RO2 protein, and we got that it shows 3.1 Newton per 7 molar dissociation constant. It is ever dissociation constant of different association and dissociation at different concentrations of RAG protein. And then this is a complex model of RAG and with PAC4 protein generated from the AlphaFold model. And this is the interfacing residue which is supposed to interact with there. And also we perform the row 1 PAC4 interaction, row 6 PAC4 interaction to compare with RAG protein, like which one is more specific to the PAC4 protein. Then I conclude my work is that HFP10 protein is very long 876 amino acid protein. So this is the alpha fold model of HFP10 protein. So it is unable, and it was not unable to be purified by E. coli system. So we purified is baculovirus system, and we trying to solve the cryo structure of HFP10 protein. And then but right now I've completed the work like this C-terminal domain of HFP10 interact with myosin proteins, myosin domain of SSC domain of myosin proteins, and then it also act with the other method of the HFP10 DHPS domain.
Some network connectivity issue looks like, na, Rutuja?
Yeah, he suddenly froze.
Yeah. Let's see if it gets better. If anything, the timing is on the conclusion slide, so he's got through the bulk of his talk. Okay, this if everyone else can hear us. Yes. Okay,
Yes.
then thank you. While Avinash is trying to join back, which we think is hopefully he'll be able to join, a good time to announce the winner of the giveaway. And the giveaway winner is Divya Jyoti Patnayak. Divya Jyoti, are you here? Divya Jyoti Patnayak. Um, he may join back again. Divya Jyoti can email us at hello@indiabioscience.org and we will ship him the physical copy of the book. So that's great. I hope somebody communicates it to Divya Jyoti that he's won. Maybe we will do that post the session. All right, the feedback form is right there. This is for the audience, not for the speakers and mentors. We will follow up with an email for that feedback form. And today afternoon, 1 to 2, Shravanti from Germany is joining us to talk about postdoc careers out of India and all that you need to consider when you're finding a lab, negotiating your uh, relocation and the kind of science you plan to do, and so on. And then in the afternoon, we have an action-packed session again, medicine and physiology, with 5 speakers and 3 mentors. All right. So look forward to seeing you all back at 1 p.m. and then at 2 p.m. for the medicine and physiology session. I don't see Avinash having come back, right, Rutuja? Do you see him?
No, he hasn't yet.
He hasn't yet. All right. What would be the best way to uh, sort of still be able to communicate the feedback from the mentors to him? Maybe Ashutosh, Salim, and Parveen, if you want to tell me the feedback, I'll note it down and email him. You know, after having given the talk, it would be a shame if he doesn't get the feedback. So you want to go first, Ashutosh?
Yeah, so I mean, certainly a lot of work and knowing how difficult it is to actually get the proteins and crystals, and so definitely a lot of work. Perhaps a feedback would be to when he's presenting to highlight what what has come out. So, for example, he has performed a lot of interaction studies between these proteins, and what is the implication from that? That's one. The other is he mentioned something about docking of these 2 proteins that we saw the BLI interactions. Then he mentioned the docking score corresponding to that. That is something which is best presented with uh, what method was used and how it was got. And so the correspondence between what he's getting in BLI and the docking score uh, may not be something which is too significant, depending on the method which was used for docking. So that is something that
Sounds good.
made it correct. Yeah. Otherwise, definitely a lot of work and very nice piece of work actually.
Hello, am I audible?
Yes. Avinash, you're back.
Okay.
Ma'am,
Great,
the
Avinash.
Wi-Fi is
Yes,
Wi-Fi
yes,
is
we get it.
down.
Don't worry, we get it. So, Avinash, that was your conclusion slide. Do you want to show that slide now? Wrap up?
No, ma'am, I connected by mobile, so the
So you
laptop
can't.
is Wi-Fi.
Okay, no worries.
So
So
that
Ashutosh was just giving you feedback, and now you can take the feedback directly from him. You may want to note it down, and you can think about it later for your work.
So Avinash, I was just saying that very nice piece of work, a lot of work actually, and I understand getting these crystals and the structures is not easy. So definitely a lot of work. I would just say that when you are presenting the interaction studies, particularly the BLI studies, it might be good to highlight in different plots which interaction you're talking about and what is the significance of that interaction that you're getting. That's first. The other is regarding the computational docking that you mentioned. I don't know. I think Avinash froze again.
Yeah.
Avinash, can you hear me?
Yeah, he froze again, Ashutosh. Looks like
Yeah.
it's a major connection. Never mind. So I got the point of docking, what method was used, and
Yeah, it might be worth mentioning that. Yeah. Okay, all right. So yeah.
that's it from your end. Great. Salim and Parveen, any feedback for Ashutosh for Avinash that I can?
Yeah.
Well, we are getting to see different parts of your institute, Avinash, as a result of this. Well, welcome back. And we'll go on to feedback from Salim and Parveen now, Avinash, just to keep with time.
Okay, ma'am.
So Avinash, it was a lot of work that you did, no doubt. Okay. So I was a bit confused, as in the title of the talk. I was hoping to see some,
Yeah.
mechanics, which in the data you did not show. So are you also pursuing that, like to really show what happens inside the pathogen?
Sir, actually, this is the starting. Like we saw the structure and mechanism, then we try to find the inhibitors, scanning inhibitors, and then popularize it. This is our idea to
Okay.
to the experiment.
Okay. But are you
So
trying
I
to
was working on that.
plan also to look at the mechanics of these
Yes, sir.
cytoskeletal elements in real time? Like, you have access to, I think, all the imaging facility there, right? So maybe you should, in addition to the structural aspects that you showed, that would also be very nice to look into. Yeah.
Okay, sir. Yes, sir.
Okay, that's it from me, Karishma. Yeah.
Yeah, thank you, Salim. And Parveen, are you there? Because Parveen's also been having some internet issues. I don't see him, right, Arushi?
He
Yeah, I
doesn't
don't
seem
see
to be
him.
there. Yeah.
I don't see him. No worries. Let's see how. Okay, I think we'll call it a day just to, protect everyone's time. And thank you, Salim. Thank you, Parveen. Thank you, Ashutosh, for your feedback. Thank you so much. Thank you to all our speakers. Thank you, Suman, Kinjal, and Avinash. Well done. Well done.
Thank
Best wishes. It takes a lot of courage to put yourself out there during your PhD. We've been there. And so hats off. You crossed multiple boundaries when you did that, multiple obstacles. So well done. Well done. It only gets better. So
Thank
really
you.
well done. Yeah. And be in touch and we'll catch up. Thank you so much to our mentors. Thanks for your time. Thanks
Thank
to
you.
Team India Bioscience. And it doesn't end. We have to be back at 1 p.m. All right. Okay. Good day. Good day.